I'm Dr. Keller, and this is Biology Level 2. In this video, I'm going to talk about a relatively new and exciting area of molecular biology, the field of molecular machines. Molecular machines are actually one of my favorite topics. When I was in graduate school, I worked on molecular machines, and so this is one of my most favorite things to talk about. So, what is a molecular machine? Well, a molecular machine is just what the name implies. It's a machine so small that it operates on the molecular level. In living things, there are literally millions of tiny molecular machines, and a living cell is just stuffed full of these guys. These tiny molecules do all the work inside the cell. Most molecular machines are made of proteins, which again are amino acid chains that fold into particular shapes. There are some molecular machines that also have pieces of RNA in them. These proteins, though, fit together to make what is called this assemblage of parts. Okay, so a molecular machine is really this assemblage of parts, and it functions just like a macroscopic machine. It transmits forces, motion, or energy from one place to another. So it does work inside a cell. So let's back up a little bit and talk about machines in general. What is a machine? Well, there are two basic categories of machines: simple machines and complex machines. A simple machine is something like a screw or a wedge or an inclined plane. A simple machine helps someone do work by lowering the amount of energy required for that work. So, for example, I'm not very massive, nor am I very strong, and it would be really difficult for me to move a chair up a flight of stairs. I wouldn't be able to generate enough force to lift the chair. However, if I used a pulley or an inclined plane, I actually would have a better chance to move the chair to a higher level, because this simple machine would do some of the work for me. It would lower the amount of energy that's required by me and lower the amount of force that I would need to move the chair up. In other words, to do physical work. Okay, so those are simple machines, and we use simple machines all the time. But another category of machines are complex machines. And a complex machine is an assemblage of different parts, many different parts, that are designed to perform a particular function. So an oven, for example, is an example of a complex machine. And an oven helps me cook dinner. It heats and cools when I ask it to. And so again, an oven is an example of a complex machine. It has many different parts inside of it. And much in the same way, a molecular machine is made of separate individual molecular parts. Okay, so a molecular machine is actually more like a complex macroscopic machine. A molecular machine can be defined as any number of molecular parts that are designed to perform a specific function, and very similar to a macroscopic complex machine. Okay, so let's take a close look at one such molecular machine, the T7 DNA polymerase. So how does this machine work? Well, in a nutshell, T7 DNA polymerase is a molecular machine that copies DNA. It has several moving parts, and it uses chemical energy to add new nucleotides. In the illustration, the T7 DNA polymerase is shown in yellow, and the DNA is in blue and red. So let's take a look at this molecular machine, a bit like an engineer who's trying to understand the design of a macroscopic machine. So let's take a look at the jobs it must do. So we have a piece of DNA, which is partly single-stranded and partly double-stranded. The DNA polymerase is attached to the place where the double strand turns into the single-stranded DNA, so it's at this junction. The job of the DNA polymerase is to create this second strand, and it does this by copying the genetic information, so it must read the A, T, C, G letters of the first strand. To do this, to create the second strand, DNA polymerase needs fuel. And so what it uses for fuel are these little monomer units called NTPs, nucleotide triphosphates, and it spits out PPIs, or pyrolytic phosphates. These monomers are the fuel that the molecular machine uses. Okay, this polymerase also needs to catalyze the polymerization reaction, so it acts like a catalyst. It's actually an enzyme. It also has to move along the DNA and straighten it out. So in other words, it needs to pull that single-stranded DNA straight as it goes. And all of this that it's doing takes energy. It must somehow use the energy of the NTPs to do all this work. And this is what makes it a motor or a machine. It has to do all of this in the presence of large, what are called thermal fluctuations. So there's heat battering this molecule around in solution. So it's moving kind of randomly, and it still has to do all of this work. It has to do it very fast, and it has to be accurate. In other words, it can make no mistakes. And so the design considerations that we have to think about when you think about a molecular machine, particularly this T7 DNA polymerase, is that first it must be able to find the junction. It must initiate the synthesis. It must be able to come on and off at the end of the job. And in the real system, it must do this on double-stranded DNA, so that means it needs to coordinate its activity with other proteins, because another protein is required to unwind the double-stranded DNA so that it will have access to the single-stranded region. So you can see there's really quite a lot going on with this little molecular machine. Okay, so I have a movie of T7 DNA polymerase. Unfortunately, I couldn't get the movie to load and actually open during the talk. So I'm going to show you what the movie is about, and then there's a second file you need to open just to see the movie. But basically what you're going to be watching is there's a piece of DNA. You can see the DNA, and it's pinned at both ends. And the T7 DNA polymerase, the molecular machine, is shown in white, and it's attached to the junction between the double-stranded and single-stranded region. And you can see there's a little yellow part of the DNA polymerase, and this yellow part is where the new nucleotides get added. And what you will see when you watch the movie of this T7 DNA polymerase is that those little that little yellow part opening and closing as it adds the nucleotides. And so there's a second video. Just click on that video and you can watch this movie. Okay, so it's really fascinating how molecular machines operate, and we can make movies of them, and we can actually study them just like we would study a macroscopic machine. So we can look at their shape and construction, just like we would look at the shape and construction of a macroscopic machine. So on the left in this image is a cartoon drawing of the polymerase and the DNA. And notice that the DNA is represented by this threaded rod, and the polymerase is represented, part of the polymerase is represented by a solid orange body. And the part of the polymerase that moves up and down, you can see is labeled hinged fingers or hinged lid, which are the fingers of the DNA, or actually of the polymerase, sorry. And this hinged lid is attached to the body of the polymerase through this little teeny spring. And so you can see there's a lot of modeling happening as we understand how this polymerase works, and we're modeling it. We're studying the shape and construction of it. Okay, so we can also look at the energies and forces that are created during the procession of the polymerase. So in other words, as the polymerase is moving down the DNA template, we can look at all of these interactions. We can look at the chemistry. We can look at the energies that are required for the chemistry to happen. We can start to study their forces. We can look at the interactions between the proteins and the NTPs, and we can look at the interactions between the finger region of the polymerase and the body. And we can do calculations, and so we can start to understand and start to study how this little molecular machine works, at a physical level. So I want to show you another molecular machine that's one of my favorites, and this is Kinesin. Kinesin is a little molecular motor or molecular machine that walks cargo, literally walks cargo from one place to another on a molecular road, which is called a microtubule. And so you can see here in the illustration that there are two of these what are called kinesin heads, and they are wrapped around, their tails are wrapped around each other. And at the end of the tail, you can see this cargo that's attached. And what happens is that these little heads move along the microtubule, and they literally walk the cargo down the tubule, the microtubule. And so I want to draw your attention to the URL, the website that's shown on the slide. And what this website is a little video, video of the molecular machine Kinesin walking down a microtubule. And it's a great little video that was put together by the Ron Vale Lab. And I want you to pause this video, or at least copy down this URL, post it into your browsers, and watch this little video of the kinesin heads moving down the microtubule. Because it's really wonderfully done. It's narrated. It'll tell you what's happening as these kinesin heads move. Okay, so there are tons of different molecular machines out there. There's just so many I can't list them all. And so there are pumps and rotors and bearings and cutters and assemblers and folders and condensers and twisters and untwisters and tweezers and on and on and on. And there is almost any type of machine function you can imagine. And probably inside cells there are many more machine functions we've never even thought about. We are just now discovering all the variety of molecular machines that are inside cells. Okay, so in summary, I'd like to say that, proteins, mainly proteins, are the ones that assemble into these large number of molecular machines. And so sometimes you get a little bit of RNA, but mainly it's proteins that assemble and create these molecular machines. In many ways, molecular machines are just like macroscopic machines. They transmit forces from one place to another, and they do work inside the cell. And because they're very similar to macroscopic machines, we can study them. We can study them at the chemical level, we can study them at the physical level, and we can even study them at the macroscopic level. In other words, we can actually take pictures of some of these machines doing their work. And so that concludes my lecture on molecular machines. And if you have any questions about Gravitas Publications or Real Science for Kids, please visit me at www.realscienceforkids.com. Thanks.